Okay, so DNA, RNA and protein synthesis. DNA controls protein synthesis. How can a single type of molecule like DNA control all the activities of a cell? The answer is very logical. All chemical reactions in cell and therefore all the cell's activities are controlled by enzymes. Enzymes are proteins. We have known DNA is a code for proteins, controlling which protein is made. Thus, DNA controls the cell's activities. Protein molecules are made up of strings of amino acids because amino acids are the monomers of proteins. The shape and behavior of a protein molecule depends on the exact sequence of these amino acids. That is its primary structure. You have learned in O levels and also in A levels is DNA controls protein structure by determining the exact order in which the amino acids join together when proteins are made in a cell. So it is true that DNA controls all the activities of the cell. That's because all the activities of the cells are controlled by enzymes. DNA controls all the activities of the cell by producing enzymes which are proteins which controls all the activities of the cell. So DNA actually makes proteins, not just any proteins, enzymes. The triplet code. As I have said, 3 nucleotides are responsible for the production of one amino acid and that they are very specific. Now, the sequence of nucleotide base in a DNA molecule is a code for the sequence of amino acids in a polypeptide. Yes. Figure 6.12 shows a very short length of DNA of a DNA molecule, just enough to code for 4 amino acids. Each amino acid needs 3 nucleotides, so 4 amino acids, 4 multiplied 3, 12 nucleotides will be there. The code is a 3-letter or triplet code. Each sequence of 3 bases stands for 1 amino acid and they are very specific. The sequence is always read in the same direction and from only one of the 2 strands of a DNA molecule, the so-called sense strand. In this case, assume that this is the lower strand in the diagram. The complementary strand is referred to as antisense strand. So, for we know that DNA contains 2 strands. Now, the strand which is read to make the amino acid is called the sense strand, and the strand which is not read, which is not used as a template to produce the amino acid is called an antisense strand. Okay, reading from the left hand side end of the lower strand in figure 6.12 is, okay, see, 3 bases coding for 1 specific amino acid. So this short piece of DNA carries instruction to the cell: make a chain of amino acid in the sequence valine, lysine, leucine and glycine. The completion of triplet code is shown in appendix 2. See, it said that the lower 5' to 3' is the sense strand, and it is always read from left to right side. Okay, not right to left, left to right side. Okay, now look at that. 12 bases, each 3 pair of nucleotides starting from the left hand side codes for 1 specific amino acid like CAA. CAA codes for valine. Okay. TTT, that is thymine. It codes for lysine. So the first 2 are valine, lysine, and like that, so on. Okay. So an example of mutation is sickle cell anemia. It is a disease. One mutation that has significant effect is the one involved in inherited blood disorder sickle cell anemia. So this disease, sickle cell anemia, is actually inherited. Hemoglobin is the red pigment in red blood cells which carries oxygen around the body. We all know. A hemoglobin molecule is made up of 4 polypeptide chains, each with one iron containing heme group in the center. So it has a quaternary structure. Two of these polypeptide chains are called alpha chains and the other two beta chains. The gene which codes for the amino acid sequence in beta polypeptides is not the same in everyone. In most people, the beta polypeptides begin with the amino acid sequence. Well, there is the sequence. This is coded from the HBA allele of the gene. So the antisense, the DNA, this is the part of the antigens from which the normal beta polypeptides are produced, has the amino acid sequence like this. You can work out the sequence of the nucleotides. You just have learned. You just need to see the dictionary, that is the DNA chart, not the dictionary, I'm sorry, the DNA chart. I will show you what is the DNA chart and how to use a DNA chart. Okay, so this is coded for HBA. HBA is how you represent it in normal allele of the gene. But in some people, the base sequence CTT is replaced with CAT and the amino acid sequence becomes. So what we can see is that the third last amino acid sequence is changed from GLU to VAL, valine. Okay, so we have known that the triplet nucleotide codes for an amino acid, and the sequence of the triplet nucleotide is specific for an amino acid. Since CTT produces GLU, but when the T in the middle is replaced with A, adenine, due to some mutation, it produces VAL. And for VAL being produced, we have also known that the sequence of amino acid is characteristic of the protein. Since VAL is produced instead of GLU at the third last chain, amino acid chain, the function of the way the new polypeptide works has been changed. This is coded from the HBS sickle cell allele of the gene. So mutation causes the allele to be changed, the sequence of the allele to be changed, for which the sequence of the amino acid is changed, for which the function of the protein is changed. This type of mutation is called substitution, because here T was substituted with A. In this case, the small difference in amino acid sequence results in genetic disease sickle cell anemia in individuals with 2 copies of HBS alleles. Now, we know that a DNA contains 2 alleles, 2 strands of DNA. If in both these strands that produce the beta polypeptides, T has been replaced by A, then acute sickle cell anemia will occur. That is not acute actually, that is the sickle cell anemia will occur and you will see symptoms. But if say one of the strand was substituted with with adenine instead of the thymine in the middle, and the other strand had CTT, then 50% of the red blood cells produced would have sickle cell anemia, forming like defected beta polypeptides, and the 50% of the other red blood cell would have the normal polypeptide. So the symptoms of sickle cell anemia wouldn't be so much severe. So if both these strands of the DNA were mutated, then a severe condition will happen. And there are also other two types of mutation, that is addition and deletion. So as we have understand, the addition causes an extra nucleotide to be added to the present strand of DNA, and deletion causes an allele to be deleted. But substitution causes only one of the amino acid to be changed. but addition and deletion causes the whole amino acid to be changed. So the polypeptide is fully, the function of the polypeptide that is produced is fully damaged. And this is also called a frame shift. Let me explain. You have understood what is substitution. Addition and deletion you will also understand, but say for example the sequence of amino genes for this is like this: 89, 89, 89. Okay, let's see is that. Yeah, so 89, 89, and thymine, thymine, thymine. Say if 89, 89, 89, if 89, 89, 89 produces say proline, and thymine, thymine, thymine produces serine. But and say there was another something like C, the next strand, that is here something like a C, which is the next strand. Okay, but if we have a, say for example, a deletion, this first 89 was deleted, what's the strand now? 89, 89, thymine, again thymine, thymine, cytosine. 89 thymine won't produce proline. Say for example, it produces valine, and thymine, cytosine won't produce serine, because thymine produce serine. So thymine, cytosine will produce, say for example, others like GLU, glutamine. So all of the amino acids have been changed. So there was triple A, triple T, and then C and something else. So 3 nucleotides triple A, 3 nucleotides triple T. But since the A was deleted, we will read from this double AT, double TC. That is 3 nucleotides. So the whole frame of the gene has been changed. This is called a frame shift. The same thing happens if there was an addition of gene. So substitution, although it is very dangerous, but it only changes one of the amino acid of the protein. But an addition mutation or a deletion mutation will produce a whole new polypeptides. So these are much dangerous than substitution. Protein synthesis. Now we will look at how the DNA, mRNA and tRNA along with ribosomes makes protein. We know that the DNA codes for the 3 triplet bases of nucleotides in a DNA codes for an amino acid, and that proteins are made in ribosomes. First of all, the sequence of DNA that is to be made that is to be used to make the protein needs to be taken to the ribosomes. They do this by copying the DNA sequence in another kind of chain of nucleotide called mRNA, messenger ribonucleic acid. We have also seen the structure of DNA and RNA. So okay, now first of all, what happens? The DNA molecule is converted into an mRNA molecule that is complementary to its strand, and then the mRNA molecule, which is complementary to the DNA molecule, is again coded in the ribosomes to make protein. Now the amino acids which are present in the cytoplasm are brought in the ribosomes by tRNA. Let's look at them in details. The code on the DNA molecule is used to determine the sequence of amino acids in the polypeptides. Figure 6.13 on page 120 to 121 describes the process in detail, but briefly the process is as follows. The first stage is called transcription. In the nucleus, a complementary copy of the code from a gene is made by building a molecule of different type of nucleic acid called messenger RNA, using one strand, the sense strand, as a template. So suppose this is the DNA, CAA TTT. It is formed into RNA. Now we have also seen how DNA forms complementary bases. Now RNA also forms complementary bases, like with cytosine on the RNA there is going to be guanine. With adenine on the DNA, on the RNA, it's gonna be thymine. And with thymine on the DNA, the RNA is going to be adenine. So from the sense strand, complementary bases join together, which are free on the cytoplasm to form the single strand of mRNA. And mRNA, RNA and DNA differs. Like DNA is deoxyribonucleic acid and RNA is ribonucleic acid. We have seen that in our first topic. Now, using what? Okay. 3 RNA nucleotides are joined together by the enzyme RNA polymerase. So, the nucleotides form complementary bases with DNA, but they need to be attached to form the single strand of RNA. This attachment is done by the enzyme RNA polymerase. This process copies the DNA code onto the mRNA molecule. Okay. Transcription of a gene begins when RNA polymerase binds to a control region of the DNA called a promoter, and ends when the enzyme has reached the terminator sequence. So, not any part of the DNA is converted to RNA. The part that starts with the promoter and ends with the terminator is transcripted on the mRNA molecule. So this process is called transcription. So transcription is the process by which the code on the DNA is transcripted on the RNA, which is going to be used to be transcribed to a protein in the ribosomes using the tRNAs. Okay. So transcription of a gene begins when the RNA polymerase binds to a control region of the DNA called a promoter and ends when the enzyme has reached the terminator sequence. Okay, so at this point the enzyme stops adding nucleotide to the growing mRNA when it has reached the terminator point. So the promoter, when the RNA polymerase binds to the promoter, it starts binding the RNAs complementary to the sequence of DNA. Okay, the hydrogen bonds holding the DNA and RNA together are broken and double-stranded DNA reforms. So first of all, definitely the DNA molecule has to unwind and unzip so that the double-stranded helical structure is broken into 2 single strands, and then the RNA molecules are formed complementary to the sequence, and also they are joined together by hydrogen bonds. But once the transcription has been completed, the hydrogen bonds holding the RNA and DNA are broken, and then the DNA again forms hydrogen bond with its other strand, the antisense strand, and also it forms into the helical structure back again. The last triplet transcribed on mRNA is one of the DNA triplet coding for stop, which is usually ATT or ATC or ACT. Okay, ATT, ATC or ACT. The next stage of protein synthesis is called translation, because this is when DNA code is translated into an amino acid. Okay, the mRNA leaves the nucleotide and attaches to a ribosome in the cytoplasm. So ribosome is in the cytoplasm, and the transcription happens in the nucleus, where the DNA of the eukaryotic, where there is the DNA, and then the mRNA goes to the cytoplasm through the nuclear pore, and in the cytoplasm the ribosomes are present, where the ribosomes use the mRNA to form the particular protein with the help of tRNA to bring the amino acids. Okay, we will see them in details. In the cytoplasm, there are molecules of transfer RNA, tRNA. These have a triplet of bases at one end and a region where an amino acid can attach to the other. The triplet of bases in one end. So each tRNA is specific to an amino acid because each 3 nucleotides in the DNA codes for a specific amino acid. So the triplet of bases, if the triplet of bases on the tRNA and triplet of bases on the mRNA matches, which is the triplet of bases on the DNA, then the transfer RNA will bring that amino acid to the ribosomes. Okay, we will see them. There are at least 20 different sorts of tRNA molecules, each with a particular triplet of bases at one end and able to attach to a specific amino acid at the other. So at the other end, it can attach to the specific amino acid. The tRNA molecules pick up their specific amino acids from the cytoplasm and bring them to the mRNA on the ribosomes. Okay. The triplet of bases, which is anticodon of each tRNA, links up with the complementary triplet, a codon, on the mRNA molecule. Okay, so new terms. What is a codon? A codon is the triplet nucleotide bases in the dRNA or mRNA. And what is an anticodon? Anticodon is the triplet bases of nucleotides in the tRNA. So when the anticodon matches with the codon of the RNA, which is the codon of the DNA, same thing, then that amino acid will be used in ribosomes to form that protein, because you know the structure of protein is very specific. I have told this many times. 2 tRNA molecules fit onto the ribosome at any one time. Yes, because one tRNA, another tRNA comes in, they forms bond, the previous tRNA leaves, a new tRNA comes and another bond is formed, and then again the previous tRNA leaves. Okay, we will see that in the diagram, but if you can imagine, it is better. This brings two amino acids side by side and a peptide bond is formed between them. Usually several ribosomes work on the same mRNA strand at the same time. They are visible using an electron microscope as polyribosomes. So suppose there is a strand of mRNA. Not just one ribosome is used, but many ribosomes work together so that large number of proteins can be formed. So they are called polyribosomes. So the base sequence of the DNA molecule determines the base sequence of the mRNA, which determines which tRNA molecules can link up with them. And also tRNA molecules needs to be complementary to the mRNA strand. So the tRNA triplet basis of nucleotide is same as the triplet basis of the DNA, and the mRNA is complementary. Okay, I will show you that. Since this type of tRNA molecule is specific just for one amino acid, this determines the sequence in which the amino acids are linked together as polypeptide molecules is made. Okay, so what I've tried to explain was that suppose there is adenine, adenine and adenine. This is the DNA. The mRNA will have thymine, thymine, thymine. This is the mRNA. And then the tRNA will have again complementary to thymine, which is adenine, adenine, adenine. This is the tRNA. So these the code of the nucleotides of tRNA is matched to DNA, and mRNA is just a messenger RNA which brings the code to the tRNA. Understand this. It means that exactly the DNA is used to make the sequence of proteins. That's why this complementary system was formed. Okay, so now let's see the diagram. Look at that. The nucleus, the purple color one, and the cytoplasm, the brown color one. And this is the nuclear pore, nuclear envelope, cytoplasm, amino acids, free amino acids are present in the cytoplasm, free tRNA are present in the cytoplasm, free mRNA, free RNA molecules are present in the cytoplasm. So what happens first? In the nucleotides, in the nucleus, part of the DNA unwinds and unzips as the hydrogen bonds between the bases break. Free activated RNA nucleotides pair up with the exposed bases of one strand only in a similar way to that shown. So the RNA strands need to be activated, that is phosphorylated. Okay, so that it has energy. As the RNA nucleotide, as the RNA nucleotides pair up with their complementary ones, their sugar phosphate groups are bonded together by RNA polymerase to form a sugar phosphate backbone. The RNA molecules are free and only one nucleotides are there, so they need to be joined together after they have formed the complementary with the DNA molecules. So that bond is formed by the enzyme RNA polymerase. The new single-stranded molecule which has formed is called messenger RNA. It leaves the nucleotide via a pore in the nucleus via a pore in the nuclear envelope. So also that mRNA and DNA molecule are now bonded by hydrogen. These hydrogen bonds also has to break. Okay, so in the cytoplasm there are free amino acids and transfer RNA. At one end of each tRNA molecule is a site to which an amino acid can bind. At the other end are three unpaired bases. Okay, look. Anticodon, which are the three unpaired bases. These pair up with their complementary messenger RNA so that they can bring the specific amino acid which is attached to this part of the tRNA. Okay, and this is called anticodon because I just explained the definition. You write it in your note copy that anticodon is the sequence of three bases nucleotides in a tRNA, and codon is the sequence of three base nucleotides in a DNA or RNA. Okay, at the other end are three unpaired bases. The base triplet is called an anticodon. Each tRNA molecule bonds with a particular amino acid under the control of a specific enzyme and with energy from ATP. Definitely for bond forming, energy from ATP is needed because bond breaking releases energy and bond forming requires energy. Okay, so each tRNA molecule bonds with a particular amino acid under the control of a specific enzyme and with the energy from ATP, and all biological reactions need enzymes, and enzymes are specific. Okay, so this is nothing to explain here. Now what happens then? Meanwhile, also in the cytoplasm, those things after the mRNA has been came out of the nucleus via the nuclear pore, those that in the tRNA, the tRNA, free tRNA and phosphorylated tRNA, this happens in the cytoplasm. Also that the RNAs are present, free RNAs. So meanwhile, also in the cytoplasm, the mRNA molecule attaches to a ribosome. Yeah, the mRNA molecules attaches to a ribosome. Ribosomes are made of ribosomal RNA and protein and contain a small and large subunit. This one is the large subunit and this one is the small subunit. Okay, 6 bases at a time are exposed to the large subunit because we just have known the point that the ribosome two tRNA molecules can be present in one ribosome. One tRNA molecules contains 3 bases of nucleotides. So because it brings one amino acid, and we learned that 3 amino acids, 3 nucleotides, specific nucleotide codes for a specific amino acid. Okay. Ribosomes are made of ribosomal RNA and protein and contain a small and large subunit. The mRNA binds to the small subunit. 6 bases at a time are exposed to the large subunit. The first 3 exposed bases or codon are always U, AUG. So AUG is the start codon. So T. Oh, and remember, uracil, not thymine. So in DNA, the start codon is thymine, adenine, and cytosine. Now also remember that in RNA there is no thymine, there is uracil. We have known this in the previous topics. A tRNA molecule with the complementary anticodon USC forms hydrogen bonds with this codon. Yeah, so tRNA forms hydrogen bonds with the mRNA codon. This tRNA molecule has the amino acid methionine attached to it. A second tRNA molecule bonds with the next 3 exposed bases. Yes, because 6 bases are exposed. The 2 amino acids are held close together and peptide bond is formed between them. This reaction is catalyzed by the enzyme peptidyl transferase, which is found in small subunit of the ribosome. Okay, and then what happens? This MET will leave, and the TR and the ribosome will go to the next part, and another amino acid will join. Okay, we will show you. The ribosome now moves along the mRNA, reading the next 3 bases on the ribosome. A third tRNA molecule brings a third amino acid, which joins to the second one, and first tRNA leaves. Okay, look at that. First parts, one tRNA with one amino acid, 2 tRNAs with 2 amino acids. Now, since they have formed peptide bonds, the ribosome goes to the next part. Look at that. This part was red, and now it went to the next part. See, it was here, now it is here. So in the next part, the TIR will obviously be here in the back because it just moved. And when the next part is read, the new tRNA molecule brought the next amino acid, and again hydrogen bonds was formed. Like this, it continues all along the mRNA until the stop codon UAA forms. So UAA prevents further translation. You need to imagine this. Okay, take time and understand. The mRNA molecule does not move. The ribosome moves along the mRNA. Okay, so now try to imagine. Now let us see the structure of the tRNA a bit. So this is the tRNA molecule. This is the amino acid attachment site. These 3 bases are always CAA. Okay, hydrogen bonds create some areas of base pairing within the single strand. Very good. And this is the anticodon. This is the anticodon site. This is where the 3 bases are specific. In this part it is not shown. Look at that. In this part there are 3 bases like this, and these are specific to one tRNA. And these bases, the different tRNA molecules contain different combination of triplet bases. Okay, and each triplet bases codes for this stands for the specific amino acid which is going to be joined here. Okay, so the part of a messenger RNA mRNA molecule. This is the part of an mRNA molecule. Very easy. No thymine, uracil, because this is an RNA, ribonucleic acid. The molecule transfer RNA, tRNA. The molecule is a single-stranded polynucleotide folded into a clover leaf shape. Transfer RNA molecules with different anticodons are recognized by different enzymes which load them with their appropriate amino acids. So different enzymes loads different amino acids in the different tRNA molecules with the different anticodons. And usually the enzymes are called peptidyl transferase. Now there are different types of peptidyl transferases. Okay, so that's it.